My name is Qiu Shuang. Everyone call me Q. I'm a fourth-year grad student from Bethany Lab Stowers Institute. Inside the cells, the DNA we get from parents first produce an intermediate messenger mRNA. mRNA is a direct template for us to produce proteins through a process called translation. During translation, the ribosomes will first go to the 5' end of the mRNA and it will translate the coding region to produce protein. This is the protein production machinery. Not only which protein you get, but also how much protein you get is really important for the cells. People usually think one mRNA only contains a single coding region inside of this mRNA for a single protein. The sequence on both sides are untranslated regions. The untranslated regions, in fact, they also contain small translated open reading frames. The small open reading frames in the 5' UTR on the upstream of the coding region is the upstream ORF. They could repress translation and thus decrease the amount of protein product. The small ORFs in the 3' UTR are downstream open reading frames. People have never paid any attention to the small ORFs in the 3' UTR. In this study, we found more than 1,000 translated DRFs in both human and zebrafish. So this is really a big group that has been ignored for a long time. The same gene that codes for the same protein in different species is the ortholog genes. For both human and zebrafish, they tend to share DRFs in ortholog genes, although the amino acid sequence for the DRF are not conserved. We find interestingly opposite to URF, the downstream open reading frames can enhance translation. If you have more DRFs, the regulatory effect will be stronger. The translation activity itself, not the product, is important. And maybe there are some mutations related with the downstream ORF can be related with human disease like cancer or neurodegeneration disease. Some of the oncogenes, they are the early drivers for the cancer evolution. And interestingly, we have found a lot of oncogenes. They also contain DRFs and they are under regulation. Maybe in some patients, we can find some mutations at the DRF itself that will change the amount of protein and finally lead to cancer or other human diseases. After I joined the lab, people inside the lab and also the bioinformatics, they are specialists in this. They really helped me to get involved in this process. So now I'm kind of half of the experiment and also half of the data analysis, which really speed me up for the new hypothesis validation and finally the paper and new result, new discoveries.